or afternoon, everyone. I'm Brian Mailloux, and I'm going to be talking about our wastewater sampling at Barnard College. And I'll be doing the first half of the talk, then Nicole Rondo will give the second half. She's been really instrumental in actually making it all happen in the past year and a half. And so it's been an interesting project. I think, right, when the pandemic started, we all wanted to see what we could do to help. And we weren't sure here, but a lot of us came together to sort of develop this wastewater sampling program at Barnard. And I want to sort of tell you the story and then start to show you some of our results. There's a lot we learned from here. It's been really exciting to be able to work with everyone at Barnard so close to home to do a lot of exciting science and make a difference. So I'm going to give you a little talk overview. We'll give a little background on why wastewater, why we're studying it. Then I'll give you the backstory, how it all came together. Then how we set up the system at Barnard, our first results, or what the results looked like, and then sort of how well we can do, how many cases can we detect. And then we sort of had right before break last year, we had Omicron come to campus. So we can sort of tell you how that happened, and we'll talk a little bit about the lessons learned. And one thing I'll try not to do is that too many bad puns. It's very easy here, so sometimes you don't even know you're doing it when you're talking about wastewater. So it's sort of, we'll see what happens, but I'll try not to do too many. Okay, so first, why wastewater? So back in May 2020, one of the first studies came out, and this was really interesting. It was a group in the Netherlands who had started looking for the SARS-CoV-2 in their sewage, in their wastewater. And they reported that you could see its presence before they saw it in the community. So it became like an early warning system. And it was a way to sort of study a larger population. So this was really exciting when this first came out. So it gave you a new way to testing. And if you remember back then, testing was really difficult to get, right? There weren't many tests available, right? There weren't many people. It was really hard to do. So if this way you could test a whole community at once, you could sort of understand the prevalence and how things were spreading. And it could be an early indicator. So this was sort of like an exciting first stage and got us thinking right from the beginning. And then in addition, later on, once that worked, people started implementing it all around. So this was in Connecticut, starting later that summer, they started looking at the wastewater also, and they could look at the wastewater and compare it to what was going on in the community. This way you could sort of get early warnings, you could tell when the community was improving. And so this became a way to sort of look at this bulk community health and try to understand what was happening. But I think what really caught the eye for colleges was, and this came out in the news, it was about August 2020, where University of Arizona says it caught an outbreak before it started. They started monitoring their dorms, right? University of Arizona has some researchers that have been really good at like looking at microbes in the environment. So they started monitoring this. They were one of the first groups, and this made the news. Like I think everyone sort of heard this idea that you could they caught an outbreak before it started, right? And this was a little bit different. This wasn't wastewater because they were looking at a building with students, and they were able to say, right, we looked at this building, we caught it before it happened, this was an exciting time. And so this sort of said, I think once this happened, I said, right, as soon as this happened, every college president wanted this technology, right? How do we protect the colleges? And so I think this was sort of the turning point where it's like, oh, we can do this. We can implement this on colleges and universities. And it was a little bit after this when we first got approached to ask if we could do this for Barnard. And how does this work? So what happens is, if you think about, if you have a building or a community, right, everyone's doing their business, going to the bathroom, and when this wastewater goes out, the waste has the virus in it. You shed the virus particles. So your waste is going to contain these virus particles. Normally now when you go you test your nose, right? You'll swab your nose, but at the same time you're shedding virus particles in other ways. And so what you can do is if you collect the waste, and I'll show you the waste is not quite like that, you can then extract the RNA from that waste, and then you can analyze it. And you can look at how much there is, and by looking at how much, you get an idea of maybe how many people are sick, And then also you can look at the sequencing, right? And like, we've all heard about this lately, like we can look at which variants are present in the waste, right? So we could watch Omicron becoming the dominant viral particle. So it's really, so it's a thing you can do where you can actually monitor a lot more people at once to see how many people are sick, or an approximate of how many sick, and also what different variants are around. So that sort of gives you the power of the wastewater sampling. And so just to summarize why we're doing this, right? Because the people infected with SARS-CoV-2 will shed even if you're asymptomatic. So if you're not, people aren't getting tested because they're asymptomatic, we can still see that. We can detect them in wastewater. They're stable in the wastewater. They stay present there, right? You can sample wastewater. Treatment plants is how people started. But what we're doing differently is you can start looking at buildings. And by looking at these buildings, you can look at, try to understand what's happening in the local population. Okay, so the backstory, how did we get this done at Barnard? Right, so I had done a lot of student-led research on groundwater quality. So these are just 3 pictures that I had from some work I was doing in Bangladesh. This was a well in Bangladesh outside of school we'd been looking at for water quality, including pathogens. Here was one of my former students, Taya. She was actually filtering water from this different well as we're looking for pathogens. And we'd regularly have students in the field doing this. So we were used to looking at water quality, etc., but we had never done it close to home. And then we'd end up, this was sort of like figures like this we'd end up with. This is a map from Bangladesh of a village. You can see the scale is about 200 meters across. The gray areas outline the village, the white are agricultural fields, the blue are canals and ponds, and every circle is a well we'd go and sample. And we were able to study, this was just in this one village, we were looking at bacterial pathogens and then also rotavirus. And we were trying to understand why some wells were positive, why some were clean, how we could keep the water clean. So we'd been thinking about this, but on a different scale and different location for a while. And then here at Barnard, one of JJ, who can't be here today, but Nicole, who's really done a lot of the work, will be talking about that side. He'd been focusing on how viruses turn on and off cancer cells. So he's in the biology department, so he's thinking about RNA and viruses in a different way, but he really had a lot of experience working with viruses, doing the molecular side of things. And so he was bringing it from the biological side, so he had this great background for sequencing, the molecular assays, etc. And so us two coming together actually makes a great team for how to analyze these viruses. And the light bulb, so how did this all come together? Well, it's hard to remember the early pandemic, and I was trying to remember how JJ and I came up with this, and I couldn't. I think we were sitting in one of the Zoom faculty meetings, and we ended up doing like Zoom texting back and forth, and we started talking about it. And we realized we really complemented each other well. And then I remember when I was coming up when school was closed, and I ran into Oliver Rose, the head plumber, and I asked him about the feasibility of getting samples. And Oliver looked at me a little weird, but he said, yeah, no, we could do that. And so that was really, once we did this, we realized we could do it, but we didn't know if we were going to do it. This is probably early. This was still probably May or June of 2020. And then that summer, with some colleagues at the medical campus, we helped them a bit on a proposal. And we thought we were done. We sort of helped them with some of the ideas, some of the things we knew how to do, and we thought we were going to be done, and that was it. We didn't become involved in the proposal. It was nice just thinking about things. But then, remember that fall, they sort of said we were going to go online until January. And the administration came back to us in fall 2020. They'd heard what we were thinking of doing, even though we didn't implement it, and they asked if we could sample campus. Could you set up a system on campus? And JJ and I said yes. We didn't really think through it very well, but we said, oh yeah, no problem, we could definitely do that. And so they came, and so we sort of developed an internal program that fall, and we started working with Oliver, who was, as I said, the head plumber, to try to figure out how we could actually set it up. And so we worked together to set things up, and we started to know we could do it. And so that fall, we sort of had to start to bring things together, because all of a sudden they said, they gave us a deadline. We need it working by January 1st, 2021. So we had a hard deadline in order to get things going. And so it was a little bit of a nervy fall to go from, yes, we could do it in theory, to actually get things set up and working together. Right? And we never set anything up like this on campus, and we didn't know if it was actually theoretically possible. Right? So how did we set up the system at Barnard? Right? The first thing we did is we hired Nicole. JJ and I were talking, we realized we were gonna need help to pull this off. And so we convinced the administration to hire Nicole. We hired her full time. And then we put out a call to students for help, and I was amazed. We had more students I could ever imagine that were willing to help. And luckily out of that, we ended up with about a dozen students who helped us throughout the process. And they're the ones who've truly helped us get through all the small steps from the day to day. And then the first thing we did is, from talking to Oliver, we thought Millstein would be the best place to set up our first sampler. It was a close building, it was the newest, the things were cleanest, so we're gonna go set up our sampling system there. And this is what it looks like in Millstein basement. In the Millstein basement, you have the pipe for all the waste heading out into the street. So if you look to the right, you can see the pipes coming down from above, and then they head out towards the street, and this is all the wastewater going out to the sewer. And what there is this what's called a U-trap here, and this U-trap, literally the pipe makes a U, so smells and water won't flow backwards. And so we were gonna sample that U-trap using pumps and tubing. That was gonna be our goal. And so what I'm gonna do in the next picture, I'm gonna zoom in on this U-trap, and I'll show you, because we were in this basement in this weird corner, the next picture is gonna be a little awkward, and I'll show you sort of how we set it all up. Right? And also this one, I have a bad pun. This one, Millstein turned out to be the first one we did. It turned out to be the hardest because everyone goes number 2 in Millstein. There's no other water in that building. So it actually the water in Millstein is disgusting. So we actually picked this was the hardest building to start from a molecular problem. And so here we go on a close-up. So here's that U-trap. Right, and so here's a piece of tubing, it comes out, and then if you can see back here on the wall, there's a pump. Here's a zoom in of the pump. And from that pump, we have the tubing that goes into a fridge, and in the fridge we keep a bottle where the samples are. So you can see here's the pump, here's the water flowing through the pump and heading into the fridge. And after you collect your sample, you get water that looks like this. I'll show you. I have a lot more pictures coming for you all. Right, here's another picture. This is in 600. The trap is in the basement, the house trap. And that trap, you can see the tubing here. It goes up to the pump pumps it, and it goes into the fridge where it keeps the sample cool. Right, we actually, on here we had Wi-Fi plugs so we can control our pumps. And I'll tell you in a second how the composite sampler works. This is the composite sampler, so you don't pump all the time. What it does, you tell it how many times per hour to pump. And so it sort of wakes up those hours and will pump you samples. We'll pump you some water into a bottle, and then you do that over the course of a day. So you get like a daily composite sample. And then here's another one. This one is in Elliott. So here's another picture. Here's the house trap going up to the pump, and then it's going from the pump down to the fridge. And here's the bottle that we're going to put into that fridge. Right? And just sort of something interesting, we have these Wi-Fi plugs, which seems simple, but having good Wi-Fi has made this system wonderful. So Joseph Urena at IMATS has helped us get Wi-Fi into all these weird basement corners, and that's really helped the system run smoothly. So you can see this was a snapshot last night at 5:44, and we had Brooks and Millstein pumping at night at that time. The other ones weren't turned on yet. So just having these pumps, right, if something's going to change, we can turn the pumps on and off from home. We can control when we get our samples, and we can check on the status to make sure everything's working. And then we track all of our samples. Every time we go to collect them, we have a survey, so we know, because we've probably collected about 1,000 of them so far. I was trying to think through the math, but we've collected a lot of samples over the year and a half, and so it's been a track every day, every time from every building has been really helpful to make sure we're keeping track of all the samples we've run. And so here, this is what it looks like when we go out in the morning. Here's us sampling. That's me, Janet, Oliver, and we're heading from 600 down to 616 to grab our samples. We have our bottles in the cooler as we go and collect them. And then here's what it looks like when you're getting a sample out of the fridge. You can see you end up with about 1 to 2 liters of wastewater that you'll be collecting each morning you go. And then here's a nice picture of holding up the wastewater. So you get, I gotta say, the first couple of days, it's tough thinking that you're carrying around wastewater, but somehow you get used to it. So you're used to seeing these bottles. You know what the different bottles are gonna look like, and if something's wrong. So you end up, so this is for about almost 2 liters of wastewater that you collect each time. And so what buildings we do, we began in Milstein, and so we got Milstein up and running, as I said, really because it was since it's the newest building, it had the cleanest system, it was the easiest to plumb, it was close with easy access, so when we were to get the first system up and running took time. And it really gave us, working with everyone, it gave us time to debug it and how to set it up. But then once we had that running, we realized when you think about the goals of your sampling, it wasn't the best because Milstein was open to the public. And so you couldn't control who was there and if there was a positive, what it actually meant. And we realized we really wanted to focus on student health, right? The goal was to make sure the students on campus were staying healthy. And so from there, we went and focused on student housing. And so I'm showing a picture of 600 here. So here's 600, and this way. But then, since we're in an urban setting, things get tricky quickly, and Nicole will talk about this. In 600, you have businesses. You have Shake Shack, Pret, Ivy League stationers, and you're really interested in student health. And this is where all of our Wi-Fi plugs came in. So we could control it so we'd only be using our samplers overnight. So we'd go from 9 o'clock at night to 9 o'clock in the morning in order to truly focus on times when students were most likely in the building and we could track their health. Okay, so the first results. And I think what we're gonna do to set up the first results, Nicole's gonna come talk to results, but part of what we did also was we had a great story that we did in the Barnard magazine and they made a video. And I think the video shows how actually all this works as well as anything. So we thought it'd be good we can look at the video and it shows the sampling, us going to the sampling and then doing the analyses. So we'll watch the video and then we'll look at the results. So let me see if I can, and then Nicole will go for the second half. So let me see if I can, let's see if it's gonna work. Ready, here we go. 3 days a week, we're going to sample wastewater from 6 buildings on Barnard's campus. We take our cooler, and we go do our rounds. I've done so many different weird sampling things over the years, it doesn't faze me anymore. So I'm used to working with pumps and tubing and water and samples and collecting them. That's what I do. But I never thought I'd be sampling the wastewater from campus.
Barnard is such a great learning environment, you get to learn while still making such big contributions to like real world things. And I think that is just such an invaluable experience.
Science is funny in that you can't really always predict where you're going to be able to find the answers. It's just really been incredible to be able to be a part of it, not just watch from the sidelines, but actually have an impact. I'm a part of this project from the moment that we pick up a sample from a building all the way to the end of the day where I collect the results and we analyze them.
We test wastewater at Barnard because it's part of a multi-pronged approach to testing that keeps our students safe and keeps us aware of the COVID status on campus. It's just another layer of protection. This isn't just messing around doing some arcane problem in the ivory tower. This is doing experiments that may help keep students safe. So it gives meaning and purpose to an academic life, which I can't think of any better reason to do work.
We've installed what are called composite samplers, and what they do is they automatically turn on and off so many times per hour. So they're sort of collecting the wastewater at regular intervals per hour. This is the first time I've met so many of these people who are just keeping the campus up and running and now they're helping us get to these corners and plumb them and set everything up.
When samples come in to the lab, first thing we do is we pasteurize it. We make it safe to handle. At some point, we get virus that's in wastewater, and we need to bust open that virus, break it open, so that its genome comes out, and we can purify and count the RNA. The bottom line at the end of the day is that we get a count of how many viruses are in a sample.
Being part of this project at Barnard has really pushed me as a scientist, and it's really exciting to me that I've been able to be so actively part of the science community and the community here at Barnard at a time when science is just so important.
This is the nice thing about Barnard. It's a small campus where everyone cares and is happy to work together. It's been a big team effort, like on so many levels.
I am so proud of what we've done as a team so far, and I am excited to keep this going as long as we need to keep it going.
Hi everyone, my name is Nicole Rondo. As Brian said, I am the research technician for this project. I also work in JD Miranda's lab. Unfortunately, he can't be here today, so I am here in his place. So one of the main parts where, after we had all of the samples set up, is it came to the lab where we kind of set up the protocol of how we were going to get from a big jug of wastewater to something we could actually analyze, being a viral RNA fragment. And so setting up this protocol, we had a couple of different options. We could do really sensitive options that took multiple days, or we could try to optimize it so it was shorter and we could still get enough virus that we could actually see if it was present or not. So what we decided to do is we have a one-day protocol that we came up with that doesn't involve any really expensive equipment. And it doesn't have any toxic chemicals, or as few as possible. And so we can go from collecting at 9 a.m. in the morning to having results ready by 5 or 6 p.m. And something else that we did with this, we were setting up this protocol, is that we actually optimized it with SARS-CoV-2. It was in an activated form that we got from the CDC, from the isolate of USA Washington 1, which we're assuming is from the first person who was infected in the United States. So that was really exciting that we were actually able to test our protocol with the virus we were looking for. And the way that we do it is that we have our large debris, so we have a big bottle, and then we filter it, so we get out the large debris, and we filter it again through a spin, which gets out the smaller debris, and then we have another filter, which filters out all the way down to bacteria, and then we filter it again to get all the way down to just the viral fragment, and from there, we can extract the RNA, which is something that we can take to do a qPCR, which is essentially what a PCR is what you do with like the nasal testing that we all are very familiar with. The letters PCR we've become very familiar with for the last year and a half. And what a PCR does is it tells you yes or no. Is it present or is it not? And a qPCR tells us exactly how much of whatever you're looking for is there. So what we were looking for, we have primers for the N1 gene and the RP. So N1 is a gene on SARS-CoV-2, whereas RP just is it's RNase-P, it's something that indicates human material. And so what we saw is that our assay can reliably detect about 100 genomes per milliliter of wastewater, giving us about like a 1% sensitivity, meaning that we could actually see about 1% of whatever virus we had in our sample. And if we look at this table, we have multiple different dorms, and we were able to see that when it is present, we're able to get down to a threshold of about 36 cycles, which there's like out of 40 in RT-qPCR. And this comes from the paper that we published of this protocol, and Lena was an undergraduate student with us at the time, and so this is her pinning up the paper that she was a part of on the board, which is very fun and something that we do on the 13th floor in Nashville. So we established this limit of detection through creating this protocol. And what we hoped was that through analyzing this wastewater, we would be able to have an early detection or warning method on campus alongside the asymptomatic testing protocol that Barnard was already doing. So students were being tested twice per week during fall spring 2021 and fall 2021. And then faculty who were teaching were also being tested once per week, and staff were being tested either weekly if you were interacting on campus, or monthly if you were remote. And we were wondering if this would give us like kind of a leg up. Would we be able to catch an outbreak like they did at University of Arizona before we had actually a positive test? And so we started with collecting 3 times a week. We had 12 to 24-hour periods. So depending on what other kind of businesses were potentially in these buildings that we were sampling, because Barnard is an urban campus, we were either sampling overnight or we were sampling an entire day. So like for 600, which Brian mentioned, has a Shake Shack and Ivy League Stationery and Pret, we were only sampling at night so that we were maximizing the amount of time that we could get that didn't have people that were a part of our testing program. But if we're looking at Milstein or we're looking at 616, those are buildings that are only populated by people who are within the testing program at Barnard. So we know whoever is positive in there, hopefully we can actually, we'll be able to match that to a positive nasal test through the testing protocol. And this is the same kind of testing protocol that the CDC recommends. And what we were seeing is that we were able to catch results within the same amount of time period as the nasal PCR was coming in at the same time. So how many cases can we detect? It kind of depends on the building environment, and it kind of complicates our data because we have people who are coming in and out of these buildings that don't belong to this testing protocol. And so it poses these like interesting challenges that we weren't quite prepared for when we were looking at our data on the back end of actually just like going the day-to-day of collecting the data and sending it on to administration. And so these commercial businesses share the same sewer line because we were sampling all the way at the very end, right before the wastewater outflows into the New York City public sewer. And so using these off hours methods, we were trying to detect positive cases that were only part of our community. And by the end of the day, so we would start at 9 a.m., we'd go, just like in that video, we'd go and pick up our bottles of wastewater, and we'd walk them back to the lab where I would go through the whole protocol that I just spoke about and put it onto the qPCR machine, wait an hour and a half, go back and get our data. I would come back up to our computer, look at the data with JJ, and then Brian created this kind of template that we would fill in each day. So as an example, this is from the very beginning of our sampling process. Brooks was our isolation dorm, so anyone who was sick was going to Brooks, so that's why it's saying that Brooks is very high. But we also had this moment where we had an outbreak in Plimpton, and we were seeing these values, and we were the first ones to see them because the nasal testing has not come back yet. So this is what, just like at the very end of the day, we send this off to admin, so they would know what's going on our end. And so this is an example of Plimpton. So kind of to orient you to this graph, it is a calendar form from January to April, and all green, anything green is just a negative, and then these like pinkish red squares are where we had a positive case. And then any of the numbers is when there was a positive nasal PCR case that was removed from the building and brought to the Brooks isolation dorm. And so what we saw is that at the very beginning, we had these like couple of days in January where we were getting these really high values, and we were like, someone must be sick here. And there was an entire suite that actually was sick. And so we were able to alert Barnard of that, and they also were getting back that information from the nasal PCR from the Broad, which has about like a day turnaround. So we were complementing each other really well in what we were able to deliver alongside the nasal PCR. And something else as we additionally looked at our data is that we saw that if there was 1 or 2 cases in a building, if there was 1 case in a building, we often couldn't detect it. So if you see later in Plimpton in these months, we have cases here or there, and we weren't often getting it. This could be because of the speed of flow in the building. This could be the amount of people who were taking a shower that day, who were just, there was just a lot of water moving, so we weren't able to catch it. But if we had 2 or more cases, we were able to catch it pretty much every single time. So that was good news, especially if we're looking at it like what an outbreak is. That's multiple cases. So if there's going to be a mass outbreak, we're going to catch it. And additionally, when people come back to the buildings, they could still be shedding. It's kind of an undetermined length of time where you shed this virus after you've recovered from the main symptoms and you've had your full isolation period. We didn't know if these people coming back were still shedding, or yes or no, and that's kind of still up for debate, and it seems to vary from person to person. And then this is another example of the 616, which is another Barnard building. This is only students who live in this building. And what we saw is that there was no really advanced warning. As I said, we really complemented the nasal PCR testing through the asymptomatic testing protocol that Barnard had. So here in January, we had these 2 students who were sick, and we picked that up. Again, here in February, someone was sick. We also had a positive case that day. Same here in March. These other cases where we had a positive test but no nasal PCR, that could be just because of this convalescent shedding. It could have been a contractor that came into the building to do some sort of maintenance, a staff member. So there are a couple of different ways that we could have seen positives without actually seeing a nasal PCR within our student population. And so what we saw is that we had a really good specificity when it came to if there was a positive wastewater test, we were often able to match it to a nasal PCR. Not 100% of the time, but most of the time, particularly if there was more than one student who was sick. And that if there was a nasal PCR test, we were able to pick that up most of the time as well. Again, it was because most of the time it was a one-off and we were able to always get that. So at the end of 2020, we had Omicron, which kind of popped up out of nowhere. Everyone was like, oh, Delta, we kind of all got over it. And then all of a sudden, everyone was sick, particularly here in New York City. At the very end towards the holiday break, everyone got sick. And I remember being in the lab and doing the very routine thing that I've been doing for a year and getting the results, and the entire thing lit up. And that was something I was just like, okay, I guess something's back, something's around. And we'd been hearing this from Europe, that there was this new variant, and it seemed as though it had arrived here in New York City. So it was being discussed in November 26th, this is around Thanksgiving, on Twitter. JJ then ordered these variant-specific qPCR primers so we could actually see if the variant that we had, or whoever was sick, we could see if they had Omicron versus Delta. Because of a particular drop of a gene called the S gene, we could determine if it had the S gene or if it didn't have the S gene. And if it didn't have the S gene, then it was one, and vice versa. So on December 10th, this is a week or so later, one of the 3 buildings was positive. We were like, okay, interesting. The next sampling period, every building was positive, which was a shock because this is the first time we had every single building positive, because we actually have been done really well at keeping the numbers low here at Barnard over the last year and a half. And so this is also when we got those primers in, we were able to see that one of the 3 positive buildings that we were able to detect with our primers contained Omicron. So it wasn't, not everyone who was sick had Omicron, but one of the 3 buildings. It was here, it was on campus. And so that was when Barnard started to recommend remote finals. Everything kind of shifted back to that kind of more quote unquote locked down version of like earlier in the spring 2021 as opposed to in the fall of 2021 on campus. And then on the 17th, again, we still had most of our buildings positive. And of the ones we were able to detect with our primers, they both were positive with Omicron. So by the end of that week, by the time everyone left for finals, for the winter vacation, Omicron was the dominant variant here, and particularly it was also the dominant variant here in New York City. So what did we learn through all of this, and continue to learn, is that if we're looking for an outbreak, which is like the main reason we were trying to do this to begin with, is that we were trying to make sure that if there was an outbreak within a dorm, which is a congregate living setting of multiple people in a suite, 6 people to 1 bathroom, it's tight quarters, we live in New York City, it's fine, that catching 2 positive cases in a building is excellent because you will be able to catch that outbreak. It was also an efficient way to sample 100 to 200 students at one go, multiple times a week, as opposed to having to do the PCR sampling, which is also very important to do. and that composite samplings, they work with limited hours, like as opposed to having to do it all day. We can still get the same results with only doing it with that 12-hour overnight. And it's best used to be identifying outbreaks as opposed to just a one-off case here and there. And that this is something that we can continue to use even as the virus evolves. So we've gone from all the way of the wild type, the very original form of SARS-CoV-2, all the way to Omicron. So even as this virus is getting smarter and making these little changes here and there, we're still able to detect it and catch it in the same way. So it takes a village. This is the interdepartmental, interstaff, interfaculty, it's just everything, all the way from the president's office, down to the environmental science department with Brian, to the biology department with JJ and I, to Oliver, who's the plumber at facilities, to all the other plumbers who help us go into all the buildings to get to the basements, which are not easily accessible. And that it's really wonderful because we were able to all work together to, really accomplish this common goal of keeping our campus safe in a community that we all really care about. And it's also really interesting science that we were able to kind of work together to create a protocol that had never been done here on our campus before. So it's exciting that we can do this kind of thing at Barnard. And that additionally, we have results within the day. So we sample at 9 a.m., we have it to admin by 5 or 6 p.m. at night. So they can make their recommendations for however we should move forward with the pandemic response team. However, we should keep our students the safest. So overall, I'd really love to thank Brian and JJ. They really were the spearheading faculty members. Oliver, who is the plumber who physically set everything up. Caroline with the pandemic response team, same with Cynthia Yang, and our wonderful students who have spent many an hour with me watching Wastewater spin because of all the filtering. Many of them have actually graduated in the class of 2021, but we still see Grace and Jenna multiple times a week. This is Grace here, Grace with the bottle that may or may not have broken today. So we have a really good time, and I've really enjoyed working alongside everybody. And with that, if anyone has any questions, I'd be happy to answer that. And Brian will come back up and answer that as well. Thanks.
Are you continuing to look at other variants? For instance, this new variant, Omicron variant? Is there something you can do, or do you have to get that information from somewhere else before you can change the experiment?
So the question is if we're looking at other variants, because as many people may have heard, there's a new subvariant of Omicron, and the cool thing about our protocol is that we're able to use the same protocol. It does not matter kind of what the variant is. We're continuing to detect it. And the cool thing with the Delta versus Omicron is that there was that deletion in the part of the genome, so we were able to see if it was one versus the other. I'm not sure if that that same thing is still present with this second version of Omicron that we're seeing. But overall, we'll still be able to detect it with the same protocol, which is great and ideal.
Yeah. Remember, we've also been doing the sequencing uptown, but that takes forever. Yeah.
So Brian was just saying that we've sent samples uptown to the medical center at Columbia University to do sequencing, but sequencing takes a minute, so it's much faster if we can have this sort of yes/no for a gene to determine one or the other, if that is present.
So you send them to be sequenced every once in a while, like at smaller points, or you just did it once? Or if somebody else is doing research on the sequencing there, and they're looking for potential material from us?
So the question is if we're sending out sequencing often, or if we're sending out samples every once in a while. So we sent it out once from our samples in spring 2021 because you have to have a good bulk of it, and sometimes we're not able to get a lot of it. And we also had the unfortunate part for a scientist, fortunate part for the community. We didn't have a lot of outbreaks in spring 2021 and not really in the fall either, which was great for the community. not great for data. So we were only able to get enough once that we sent up, and they have the sequencers and the ability to do that. We don't have any sequencers here on Barnard's campus. Those are big, expensive machines. They're very fancy.
Question from the virtual audience.
Yes.
Okay, so considering the safety hazards to you performing this amazing experiments, did you get a lot of no from Barnard to even launch this program?
I think that'd be a Brian answer.
No, that's a good question. I think in the beginning, there was a little bit of no, but I think as soon as that, like when people started to see the power of it, there was a push to get it going quicker, right? There was a push to try to get it implemented, sort of that in that fall 2020, because they realized that you could use it to help protect the community. So I think there's actually been a push to like sort of be helpful. It's been amazing how supportive everyone from like the president's office to then within all the staff, even like we've gone to more places around campus than I'd ever seen before. And even like when you have the students with us, they're shocked. They're like, there's something down here. And so we're going to corners of campus that are, that most students and faculty would never see. So there's been support like across the board from everyone to get it working. And then and then we had to get Wi-Fi into these corners. So they were supportive to help us get things rewired. So there's been amazing support like across the board to get everything up and running. I think once everyone realized how helpful it could be, like everyone's really pitched in and helped. There. Are you? Okay. Oh yeah, what's up?
Somebody in the community, like a staff person, so I want to thank you guys for doing this. I mean, really, it's great work, and it's very valuable to be here when this kind of work is supported.
We just got to thank you from the audience. We always appreciate that. Thank you. Sorry. Okay. Cool. So I think we're going to wrap up there. So thank you everyone for coming, and it's been fun doing this.